This is a quick recording to go through how to complete your experimental write-up for the Is Any Sugar Good Enough experiment. Some students are away, so we're just doing this so that everyone can keep up with the work and know what all of this was about. Essentially, students were given the challenge to design an experiment that showed whether or not yeast had a preference for sugar when they were respiring. And we've gone ahead and learned a bit about respiration, and we know that respiration is where an organism actually takes sugar, normally in the form of glucose, but not always, and uses that sugar in respiration reactions. And it usually takes oxygen and breaks that sugar up and gets the energy out of the bonds within that sugar, and it makes water and carbon dioxide and recharges ATP. Now, as they've also learnt, when oxygen is absent, or in the case of many microorganisms, they don't respire with oxygen. They actually take that out of the equation and they do something called either lactic acid fermentation or alcoholic fermentation, which means they break up the sugar without oxygen. It's anaerobic, that's what without oxygen means. And they break the sugar down into either lactic acid or alcohol, and in the case of yeast, alcohol, plus the lovely little byproduct of carbon dioxide. So we had to design an experiment where we kept as many of our variables the same and controlled so that it was fair, and so that we could compare how yeast respired with different types of sugar. And students had to come up with this. Here's a picture of the basic design of the experiment. Here's the students controlling the variable of temperature, because you had to keep everything at the same temperature to make sure that it was all, because temperature affects the rate of respiration along with many other things. And they had to figure out a way to measure the carbon dioxide output, and this is what they've come up with, these balloons here. And we decided to measure the volume of these balloons using a mathematical equation to find the volume of a sphere. And in order to do that, you needed to know the radius both horizontally, both ways, and vertically. So in the background here we have these rulers here, and we zoomed in on these photographs to actually take measurements of the balloons so that the measurements were essentially all at the same time. That's another way to control variables. And that way we were able to then calculate the volume of these balloons as best as we could, given a good photograph. So let's look at what we found out. I gave you this information here to say a little bit about what respiration is, and it talks about the different types of sugars that we have. Well, three to be really. The most common basic form of sugar is glucose, and here's a little picture of it here. Now, glucose exists as what we call a monomer or a monosaccharide, which means it's one little unit like this. And here's a picture of fructose, which is actually quite similar to glucose chemically, but it has some differences. Okay, you can see that the little groups here on fructose are different from those same groups on glucose. But they're both monosaccharides, single units like this, of 6 carbons. Now, most of the time, sugars in real life don't just exist as these monosaccharides. They join together into what we call disaccharides or polysaccharides. In other words, chains of sugars. This is a disaccharide here because there's 2 pieces joined together. That's called sucrose. That's actually the table sugar that you have normally and you eat it every day. And it's made out of 1 unit of glucose joined up to 1 unit of fructose. So here's another picture to help you understand what sucrose looks like. Sucrose is quite literally a glucose linked to a fructose. And there are other types of polysaccharides that you know of in everyday life. Starch, any type of starch like potato starch, wheat, all of those are polysaccharides. In other words, units of these tiny monosaccharides joined together in long chains to make a polysaccharide, which you normally call starch. And they're in all of our different foods. Now, the research is simply this. When it comes to yeast, they need to be able to use a monosaccharide in their respiration reaction. If it's anything bigger than that, they literally have to cut it up and chew it first before they can use it in their reaction. So yeasts are quite happy to eat these monosaccharides and use them in respiration. The theory is, though, that glucose being the simplest sugar, because it's only got one of these little chains up here, that it would be the one that is most likely to be utilized by yeast the easiest. But we're going to have a look at our results and see that. Anything else that's bigger like this, a yeast or anything, they can't utilize it straight away. Neither can you. When you eat starches and stuff, you can't just digest them and use them for respiration. Your body has to chop them up into its individual monosaccharide units, and then it can put them into the respiration reaction. So the theory is basically, do these monosaccharides here, do they get used in respiration better and quicker than the disaccharide that we're going to be feeding our yeast, sucrose? And have a look at the results. Now we're going to be filling this in class. This was part of our planning of how we controlled our variables, so don't worry about trying to fill that out at home. We'll do that under exam conditions. Here are our results, and as you can see, we had glucose, fructose, sucrose, and the control, which had no sugar in it whatsoever. You have to now take those results and interpret them and try and answer the following questions based on the theory that I've just talked to you about. Now, notice that the control here actually does have some gas production. How is it going to be doing that if we didn't give it any sugar? Well, any living organism, including you have small amounts of chemicals in you, stored energy, like in humans, fat, but yeasts don't always store fat. They can store sugars inside their cells so that if they are found to be without food for a while, they can still keep living and they can still respire by using their stored energy. And that's exactly what's happened here, that the yeast already had something inside them ready to use for respiration if they needed it. After that, we had sucrose. Okay, funny little shape we've got here to our graph. You'll have to try and talk about that and why that's happened, what you think's happened there. Then glucose, and then fructose. Now, when you come and look at that graph, we have to answer a few questions, and in science, it's very specific how you approach setting the scene for your experiment, talking about the data, and then discussing and analysing it further. The very first thing you have to do is tell the reader what you actually tested, because we didn't just test any sugar, we tested 3 specific sugars and a control. We tested glucose, sucrose, and fructose. So when you describe your results, the very first thing you'll do is describe the types of sugars that we investigated and explain the 3 different ones that were in our groups. The next thing you have to do is pretend that you're talking to someone who cannot see that graph, and you have to describe it in detail for them. So you have to describe the results not only of just the general results and the general which came first, but very specifically how much they produced or how much CO2 they produced over the 15-minute period, right down to the specific amounts. So I would start off by saying over a 15-minute period, the control actually produced about 200 centimeters cubed of carbon dioxide, even though it had been given no sugar to actually use for respiration. Then you'll talk about how much sucrose produced, how much glucose did, and fructose. And that literally paints a picture for someone who cannot see that diagram there and helps them to understand what it looks like. The next thing you have to do is discuss the general trend in your results. So in this case, we'll be talking which one was the most preferred sugar, down to which one was the least, and comparing them to each other in more detail. So you always start off with the general trend of which sugar was preferred down to the least, or the other way around, and then you have to be very specific and try and compare them. Okay, and when I mean compare, it's not just which was preferred, but literally you're looking at comparing how many times more popular it was. So if you look at sucrose here, that's about 600 there on the scale, and glucose up here would be about 1200, which is literally double the value of the sucrose here. So when we compare it, we don't just say it's bigger, we literally say it was double the volume of CO2 was produced in the glucose compared to the sucrose. And then you can also compare mathematically the fructose to the sucrose, because remember, this is our disaccharide down here, the one that we theoretically know should have to be chewed up before it can be used in respiration. And these two here are our monosaccharides, which are the smaller ones that don't actually need to be chewed up first. So theoretically they should be faster. You're now having to compare, yes, they're faster, how much, and do it mathematically, telling me roughly how many times bigger, or if you do it the other way, smaller, they are in the CO2 production. And the last thing you have to do is talk to me about, can you propose an explanation? I shouldn't say why they do not match the current theory, because that's incorrect in this right up here. Do the results match the theory? And if so, how? And if not, why do you think so? Because we were, in the theory, it was talking about disaccharides and monosaccharides and which one would be easier for respiration to use. But we also had the theory of glucose as the simplest sugar would be the easiest for yeast to use for respiration. Was that the case? Does that data support those theories? Or does it suggest something else? You actually have to try and think that up yourself and answer that there. There. Now, last but not least, this was supposed to be a fair test. Do you think it was as fair as it could be, or can you improve on it? And specifically, how would you improve to get better quality data for the future? So here is where you pull apart your methods, the way you measured it, the way you controlled your variables, talk about what you would change next time to make a fairer test, and also very specifically, is there a better way that you would not only control your variables, but how you would measure the respiration rate as well? Can you think of ways that you do it better for, can't stress it enough, for better quality data? Because that's what a true scientist is always looking for. How to improve the data to make sure that it has less error in it, and make sure that we're definitely happy with the analysis and the information we've been able to pull out of it.